Dear students, today I will discuss about meiosis, and this is for our course Cell and Molecular Biology, and code is BBBMBT1002. This slide is showing how the process of meiosis or cell division, uh, meiosis is taking place. We can see how the tetrads are separating, the chromatids are moving to both the sides, and this is actually Anaphase 1. This slide is showing Anaphase. This part is Anaphase 1 when the chromatids start moving to both the poles, and uh, so this is how this process of meiosis is taking. When they are in the center, they are known as Metaphase 1, and now again they are separating and moving to both the poles, and this is Anaphase 1. Now let us come to the facts about meiosis. Meiosis, it is preceded by interphase, which includes chromosomal replication. So, we have earlier studied in mitosis also we have studied that chromosomes, they replicate during S phase of cell cycle. So, they synthesize their own replica. Now, two meiotic divisions take place. One is meiosis 1 and second is meiosis 2. So it has got two segments. This meiosis division has got two parts, meiosis 1 and meiosis 2. Now, meiosis is known as the reduction division because here the original cell, which is actually 2n or diploid, we have earlier I have already explained you that in human beings we have 23 pairs of chromosomes, 22 pairs autosomes, and one pair is sex chromosomes. So the cell is it's 22 pairs, means cell is diploid. Each chromosome has its own partner also. Now, four daughter cells are produced, which are monoploid or haploid. So, in the process of meiosis, the four haploid cells are formed, haploid or monoploid cells are formed, and they are the number of chromosome is 1n. So, in the beginning, it is 2n, but after the cell division, four cells are formed and we get one n of chromosomes. Daughter cells contain half the number of chromosomes as the original cell. So original cell has 2n, but here the number of chromosome is 1n. So this cell division or meiosis takes place in gametes, or that is eggs and sperms. So in the sex cells, the division of cell is actually meiosis, and the diploid cell divides into four haploid cells. Now, this occurs in the testis in the male, and this process is known as spermatogenesis, and it occurs in ovaries in females. Here it is known as oogenesis. Now, why do we need meiosis? Why meiosis is so important? It is the fundamental basis of sexual reproduction. Two haploid or 1n gametes are brought together through fertilization to form diploid or 2n zygote. That is, this is the egg which is having 1n number of chromosome, and these are the sperms. They also have one n number. That is, the number is haploid or 1N, and after the fusion they form the zygote, which has got 2N number, and this 2N zygote then multiply mitotically to several cells having the number 2N, so as to develop into the body of the embryo. Now, again, this is the diagrammatic presentation showing fertilization. This is egg cell having haploid number, and again these are the sperms which are also this is the sperm which is also having the haploid number. As for example, in this case, suppose it has got 3 chromosomes in the sperm and 3 are in the eggs, and they both of them fuse together to form a zygote which has got diploid number, and so it has got 2N or diploid. So 3 plus 3 is equal to 6, and this is known as the zygote. Now, replication of chromosomes. This is chromosome. Replication is the process of duplicating a chromosome. It occurs prior to division, and replicated copies are called sister chromatids, and they are held together at centromere. Thus, this is the diagrammatic presentation of the cell cycle. This is showing the interphase stage, and I have explained you earlier also, when I have explained you about the cell cycle, that during the cell division is followed by the G1 phase, when the cell content is increased, cell metabolizes and grows. Then this is followed by the S phase, when cell actually starts preparing itself for the cell division. And so first step in this is the replication of the DNA. DNA replicates and synthesizes its own replica. So the replica which is formed is known as, then they are called two sister chromatids. I'll show you here. And then this is followed by G2 phase. G2 phase, we know, is the phase when the accessories which are required for cell division are synthesized, like the centrioles. It divides into two, and then formation of astral fibers and all those things. These things take place during the G2 phase, and this is followed by the cell division. So we can see here, this is showing the unreplicated chromosome, and during S phase, which is this, the DNA synthesis takes place and condensation takes place, and we get two sister chromatids, two identical sister chromatids. This chromosome has synthesized its replica, and these are the two sister chromatids, and which are connected together at centromere. This is the centromere. Now let us come to next slide. Now, here we see, this is the diagrammatic presentation of which is explaining meiosis. Two parts. This diagram is showing that it is two-part cell division. This is the diploid cell having which is the normal diploid cell. Now, during meiosis, first stage is meiosis 1, when the homologous chromosomes separate here and here. So the homologous of this chromosome is this. I think I have explained you earlier that let us come to human beings. There are 22 pairs of autosomes and one pair of sex chromosomes. So the pair means both the chromosomes are homologous. So the homologous chromosomes separate this and this. So still they are having the diploid number. But during meiosis 2, where the sister chromatids separate, this chromatid and this chromatid, both of them will separate. So it will divide into 4 cells. 1, 2, 3, 4, and will have, naturally it will have the haploid number. One chromatid each of each, each chromosome. So the number will become haploid. Now, this is again explaining the same thing. Meiosis 1 is a reduction division, and these are the different phases. This is nucleus, and these are the centrioles. Now, what happens during early prophase 1, or when the early prophase 1? Now, what happens during the S phase? Already the DNA has synthesized its replica, and each chromosome has 2 sister chromatids. Now, this is prophase, this is early prophase 1, and this is late prophase 1. See, at late prophase 1, what happens? The spindle becomes clear and centriole divide. See, here the centriole have divided, but now they move to the 2 poles at late prophase stage, and these spindle fibers have start developing, and they then join, fuse together, and these chromosomes, they arrange themselves in the center, and this stage when the chromosomes arrange in the center is known as metaphase 1. Now, this is the stage called Anaphase 1, when these chromatids starts moving towards the both poles. So this and this side. So they separate, and this stage is known as telophase. So what happens during telophase? Again, during the nuclear membrane, which during the prophase is gone, it becomes invisible, again appear. The centriole also move to the end, and cell starts dividing at the center also. So this is telophase stage. Now let us discuss the prophase 1 in details. Prophase 1 is a very significant, we can stage of the meiosis. So these are the different stages. What happens? This figure is showing the early prophase. Now, what happens? Homologous pairs, they start synthesizing they have already synthesized their replica during the S phase. They arrange themselves like this. They start condensing, and they become clear. And what happens during prophase 1? The crossing over takes place. Crossing over is the phenomena when the two sister, both the two chromosomes or homologous chromosomes come close to each other, and they form a tetrad. See, these are the two homologous chromosomes, and each is showing 2 because they have already synthesized their replica, or so that they are known as now chromatid. These two are chromatid and these two are chromatid. So what happens during the crossing over or late prophase 1? These two homologous chromosomes come together and attach themselves at the point, and the process of this is known as centromere, and this process when they form a pair, This is known as tetrad because we get four chromatids are at the same, are attached by the centromere. Now, what happens? The phenomena of crossing over takes place, and this phenomena nature has what happens, this chromosome, this chromatids pair with this with this, and the exchange of genetic material takes place, exchange of genes takes place. Some part of this goes to this and this to this chromatid. So this is known as crossing over, and nature has uh, provided this generated, or we can say this nature has designed this phenomena for getting better genetic combinations. So here the genetic combination of the two chromatids takes place, this with this and this with this, and so this phenomena is known as crossing over. So what happens during late prophase? During late prophase, chromosomes condense, spindle is formed, and nuclear envelope fragments. मतलब nuclear envelope, we have till now studied that nucleus is separated from rest of the cytoplasm by the nuclear membrane, which is porous, and this nuclear membrane then disappears. So during cell division, both nuclear membrane and also nucleolus, both these things, they disappear. Now this is the diagrammatic presentation of crossing over. And what happens during crossing over? See, homologous chromosomes in a tetrad cross over each other. See, these two are homologous chromosomes. So this chromatid crosses over with this chromatid of the other homologous chromosomes, and this phenomena is known as synapsis, or pairing of homologous chromosome. Now this is the chromosome having two chromatids from the paternal, and these are maternal. So the chromatid from the paternal chromosome and this chromatid from the maternal chromosome, they pair and cross their genetic material between the two. So this part of the segment comes to this, and this part from the this was father, so this has gone to this. So this produces, so the why nature has designed this phenomena of crossing over, because so as to produce genetic recombination in the offspring. So this is a very important point. So why this phenomena of crossing over is taking place? For getting better genetic recombination in the offspring. So I think this must be very clear. This chromosome is from father, this is from mother. But they are homologous chromosomes, and they come close to each other, and this chromatid from father and this from mother, they cross over each other. Some of the genes from this mother comes to father, the chromatid which is from father, and the some of from father goes to the mother side. So the phenomena of crossing over is taking place in this. This is clearing it. This is again showing the crossing over. See, these are the two homologous. One is from father and one is from mother. How they are coupling or pairing, and then resulting into transfer of some of the genes from mother's chromatid to father and father's from the mother, and this, and see, this is the phenomenon, and later on they separate. So crossing over multiplies the already huge number of different gametes types produced by independent assortment. So now metaphase 1 and Anaphase 1. So in the case of meiosis, Metaphase 1 is the Prophase 1 is very important, because during Prophase 1 the phenomena of crossing over takes place, and that is followed by then metaphase. A metaphase, we know, as usual, it is the process when all the chromatids or chromosomes arrange themselves in the equatorial region of this spindle which is formed. Now homologous pair of the chromosomes align themselves along the equator of the cell. And during anaphase, what happens? The homologous pairs separate and move to opposite poles. During anaphase, the movement to opposite poles have started. You can see. So homologs separate and move to opposite poles. Sister chromatids remain attached at their centromeres. This, see, with the, now these fibers are known as kinetochores, which attach to the centromere. So they start moving towards the poles. Now this is telophase 1. What happens during telophase 1? The chromosomes have moved towards the two poles. Nuclear membrane starts reappearing and spindle disappears, and this is even nucleolus gradually starts forming, and cytokinesis. Then this phenomena is followed by the process of cytokinesis. Now the telophase is followed by cytokinesis. Cytokinesis is the division of the cytoplasm. So this is cytokinesis. Now this cytokinesis divides this into the two cells. This is again the diagrammatic presentation of the meiosis 2. Only one homologous of each chromosome is present in the cell. I think this is known to everybody. Sister chromatids carry identical genetic information. Now, meiosis 2 produces gametes with one copy of each chromosome and thus one copy of each gene. Now, this is showing the whole process, this diagram. So, meiosis, this is showing the meiosis 2 also. So, what is happening? After the meiosis 1, the two cells are formed, but having the, still the number is diploid. Now, what happens? Then this is followed by the metaphase 2. During metaphase, as we know, the chromosomes arrange themselves in the equatorial region, and see, this is metaphase 2. This is followed by anaphase 2. When the sister chromatids separate, one moves to this end, another moves to this end. So now this, which was this, the two chromatids were together and were having the 2n number, these two cells were having 2n number, have separated, and the chromatid, one of the chromatid moved to this end, another to this. Now the number becomes now 1n, because both the chromatids have separated, and they form two different cells. This cell, another cell. Now this is one cell, this is another. So after meiosis 1, these two cells were formed, and this phenomena is then followed by the meiosis 2 division. And what happens during meiosis 2? These two cells, in these two cells, the meiosis 2 starts taking place. Cell again arrange themselves in the equatorial region, and This is known as metaphase 2, and during anaphase 2, the chromatids separate and starts moving towards the 2n. Now each chromatid is having 1n number of chromosomes. So here this cell will form one, this will form one cell, this is another. So this is having 1n number of chromosome, and this is also having 1n number of chromosome, and this is telophase. So what happens during telophase? The genetic materials separate and move towards the pole, and this is followed by the cytokinesis. During the telophase, gradually the nuclear membrane also starts reappearing, and then slightly towards later end of telophase, the nucleolus also start appearing, and thus and this is followed by cytokinesis, that is the division of the cytoplasm, and thus once the cytoplasm has taken place, we get 4 cells. So one cell has divided into 4 cells, and having the cell was earlier 2n, or having 2n number of chromosomes, now becomes 1n, or having haploid number of chromosomes, and they so we get 4 identical haploid cells. And these now, so it's very clear that this phenomena or meiosis takes place in the sex cells in eggs and in sperms, so that this 1n number becomes 2n once the union of egg takes place with the sperm, so as to form a zygote which has got 2n, and zygotes then divide mitotically several times to form an embryo which is having 2n. Now, so prophase 2 and metaphase 2, let us see what happens during prophase 2 and metaphase 2. See, during prophase 2, the nuclear envelope fragments, it is not visible, spindle is formed, and during metaphase 2, what happens? Chromosome align along the equator of the cell. This is anaphase 2. This slide is also a diagrammatic presentation of the same thing. Anaphase 2, what is happening? Now this is the one chromatid, and this is another chromatid, so having 1n number. This cell will also have 1n number, this will also have 1n, this will also have 1n, this will also have 1n number. This is how the sister chromatids separate and move to opposite poles. Now in telophase 2, is again the nuclear membrane starts gradually appearing. Slightly nucleolus also faintly visible. It is not shown here, but faintly starts coming. And this telophase 2 is followed by the phenomena of cytokinesis, when the cytokinesis means division of the cytoplasm. So ultimately 4 cells are formed which are having 1n number of chromosomes. So results of the meiosis. What is the result of the meiosis? These are the 4 cells. This is present in gametes, or this phenomena occurs in gametes, that is in egg and sperm. 4 haploid cells with 1 copy of each chromosomes are formed. 1 allele of each gene is there. Different combinations of allele for different genes along with the chromosome takes place, because this has already, the combination recombination has already taken place during the phenomena of crossing over, which occurs during the prophase of the cell which are having, or gametes which are having the process of meiosis. So this slide is showing the phenomena of the comparison of mitosis and meiosis. The number of division in mitosis is 1. In meiosis, it is 2. Number of daughter cells formed in mitosis are 2, but in the case of meiosis, it is 4. The genetical identity. In case of mitosis, yes, both two cells are genetically identical, but in the case of 4 cells which are formed in the gametes, or where meiosis has taken place, the genetical identical is not identity is genetically all the 4 cells are not genetically identical. They are different from each other because recombination has taken place. And even one thing I just want to tell you, in the case of the sex cell, XX and XY combination can take place during the formation of zygote. So what happens in the case of the sex chromosomes, what happens? Either X and Y will separate. So X will form X will form the one set of X will be one set will having X cells and another set will having Y cells. So now same chromosome is same as parent. Chromosomal status is same as parent, but here they are half of the parent. Somatic cells, they form the somatic cells, and meiosis, they form the germ cells. Mitosis can take place throughout the life, but meiosis takes place during the sexual maturity. They occur after the sexual maturity only, the meiosis takes place. Now the role of mitosis is in growth and repair. So mitosis goes on in our body throughout the life, for the growth of the body and also for repair. Whenever there is some damaged tissue, that is the cell there the damaged tissue is replaced by the growing cells, where the mitosis takes place and they repair the tissue. But the meiosis takes place only during the sexual reproduction. Now, these are the different references mm, which you can follow. So this was all about the meiosis. So we have discussed about mitosis, which the role of which is there in tissue repair and also growth of our body. Secondly, the second phenomena is the second type of cell division is meiosis. Meiosis occurs in the case of the sex cells or in gametes. And so in meiosis, one cell divides into 4 cells, and which are genetically different. The 4 are not the genetic makeup of all the 4 cells is not same, but in the case of mitosis, the genetic makeup of all the both the 2 cells are same. So this was all about mitosis and meiosis, both the process of cell division.